name is Ryan, and I'm going to talk to you today about our hackathon project, AI-powered sequence-to-function Wikipedia for longevity research. This is our submission. Our team is West Coast Vectors. That's Felix, Matthew, Zach, and myself. All right, let's get started. So what's the problem? Critical knowledge is trapped in unstructured text. Vast amounts of proteins sequence-to-function data that's vital for longevity research is buried in millions of scientific papers. Manual extraction is slow, expensive, and unscalable. This bottleneck hinders discovery of new therapeutic targets and the design of interventions. Our solution is an AI pipeline to automate knowledge extraction and synthesis. We build an automated RAG system that reads papers, extracts key sequence function relationships for the CC chemokine receptor family. The pipeline creates a queryable face vector store linking protein variants domains to phenotypic outcomes related to inflammation. The pipeline finally synthesizes structured Wikipedia-style summary articles for researchers to access via a next.js web server. The technical architecture has two phases. There's an offline pipeline that builds a knowledge base and articles in face, and then an online serving system that serves those articles to the website. The back-end pipeline has several key stages. Data is fetched from structural databases such as Uniprot, and the full text and abstract data is then extracted from Europe PMC. And then we're saving the paper text and metadata locally as JSON files. And then we're doing the indexing and embedding process, converting those papers into manageable chunks with Llama Index, and then using Nebius to generate semantic vector embeddings for each chunk. Then storing those embeddings in text in a searchable local face vector database. And then doing the knowledge extracting, retrieving the most relevant, say top K text chunks from face based on a protein query, and then using Nebius LLM with a scientist persona to extract relevant structured facts as JSON from the retrieved text. And finally, another Nebius call with a writer persona to take those facts and write the web page. Yeah, we can have a quick demo. So the backend fast API, we can just look at quickly here. Here's an example endpoint where you query with a protein name and some of the data scraping is performed. So that gives you a flavor of that. And then yes, the final website articles. So here is the ApoE article. We have the amino acid sequence structure preview, which looks nice. And then here's the data that was gathered. So for ApoE, we have an overview. We have a sequence to function table with the interval, modification, function effect, longevity effect, evidence, and citation. So here we have several results for ApoE, and then some notes at the bottom about ApoE, giving some more details. And then finally, here's the CCR1 article as well, similar in structure, pulling multiple citations that are longevity relevant for this particular protein. Okay, and so that's the demo. And then challenges, yeah, so the embedding vectors was slow and time consuming. There's challenges with extraction accuracy with the manuscript data. And yeah, we'd love to, in the future, expand coverage to all kinds of longevity-related protein targets, integrate fast API with LangChain potentially, and deploy the face database and API into some cloud server for scalability. Here's our tech stack that we used. And yeah, conclusion. So we built an automated pipeline using fast API face and Nebius to unlock sequence to function knowledge for the CC chemokine receptor family. And this is accelerating longevity research on inflammation, providing reusable framework for automated knowledge extraction. So please visit our live demo and check out our GitHub repository. Thank you so much for your time.